All right, I'm gonna go ahead and get started. Please do let me know if you have any questions as I go. Yeah, I'm just gonna jump right in. This little tutorial is about browsing, filtering, and downloading data from the cell painting gallery. Um, Hi, I'm Erin. Got my email on the slide. You are always welcome to email me, ask questions at any point. Yeah, at any point after this tutorial. Um, yeah, the cell painting gallery. Just want to, acknowledge lots of hard work from other people before being the one to talk myself. The cell painting gallery is a joint project in the imaging platform between both labs, the Simony lab and the Carpenter Singh lab. We are the imaging platform at the Broad Institute of MIT and Harvard. Um, the cell painting gallery has been really driven primarily by Shantanu Singh and me. Hi, I'm Erin Weisbart. I'm a senior staff scientist in the Simony lab, the lead image analyst in the lab, sort of the self-appointed lead curator of the cell painting gallery. And again, this is my email address here, so you are welcome to get a hold of me at any point. Yeah, I'm going to give a tiny bit of background. Hopefully you came to this session already having some idea of what this is about, but just to catch everybody up, in case there's folks new to the cell painting assay, it is a specific assay, the most popular image-based or morphological profiling assay where 5 standard fluorescent channels on any generic microscope are used to image 6 different stains that stain 8 different cellular organelles or compartments. And so this is an example of what some cell painting images look like. Um, the process of morphological profiling is we can start with any cell line, healthy or diseased. We can add perturbations, drug perturbation, genetic perturbation. In a high throughput, high content manner, the cells are fixed and stained, and we do image analysis and feature extraction. So we are segmenting nuclei and cell bodies, and then making thousands of measurements in the 5 channels in each extracted or each segmented compartment to get thousands of features where we have single cell data where these morphological profiles at the single cell level, where each column then represents a different morphological feature, and you can think of this as comparable to any omics data. So whatever sort of analyses of, for example, here clustering that you can do with any omics data, you can do on morphological profiling data, cell painting data. Um, so this is a sort of big picture overview. And so what I really wanted to point out about this is that means that cell painting data really has 2 major categories that we need to think of when we're browsing through data and figuring out what to download. There is image data, starting from the raw images off the microscope. And there's numerical data. And the numerical data can be at different levels. It can be raw measurements as they are made directly from the images. It can be all the way through profiles where we have aggregated all the single cell measurements into a per perturbation profile that is feature selected and QC controlled. And, so lots of processing can happen. We have sort of these 2 major bins, image data versus numerical data. And the vast majority of the data in the cell painting gallery, the extraction of image data to numerical data has been done by Cell Profiler, sort of classic image analysis algorithms, classic feature extraction. But we do have more and more data coming in that also uses deep learning features, and we do have organization for that in the gallery. but the gallery, yeah, but most of the data that we have currently is classic techniques using Cell Profiler to get the morphological data. So now let's actually talk about the gallery. So the Cell Painting Gallery is hosted on AWS's Registry of Open Data. So many thanks to AWS for hosting this data so that we don't have to pay to host it. I'll just point out here that if you do use data from the Cell Painting Gallery and publish, please, we have citation guidelines in our documentation that we'll dive into, but I just want to remind everybody to please also acknowledge AWS for paying to host this data. So the data is in an S3 bucket hosted by AWS as part of this open data program they have. And the gallery itself currently has 19 complete data sets. We have 22 data sets that are in progress, either in progress of uploading or in progress of acquisition or analysis, somewhere in the workflow. So lots more data coming in. It's about 700 terabytes right now, but clearly with more data sets coming in than there currently are complete, you know that this is going to get larger. And I also want to say that we welcome contributions. So most of the data is data that we have generated directly with a partner or collaborator. But now that the gallery is fully public, that is not universally true, and we absolutely welcome contributions of data. So if you have cell painting data that you or a collaborator are generating and would like to have made publicly available, again, drop me an email, let us know. We would love to get your data into the gallery as well. So just some of the data sets in the gallery, as I said, we have lots of data. And again, the data can be compound data, it can be genetic data, overexpression or knockdown, it can have dose response, it can be variants on the cell painting assay, specific for lipocytic cells, in a pooled format. We have historical data all the way from the very first published description of the cell painting assay all the way through the biggest flagship JUMP data set, even into data sets that are currently, in progress. So a full range. So with all of this data, how do we even figure out what we want? Well, we go to our landing page for the cell painting gallery. So our home landing page is it's a GitHub repository, but if you are not comfortable with GitHub, you don't need to be able to do anything in GitHub. It's just this landing page. And the URL is github.com/Broad Institute/cellpainting-gallery. And here you will find the most important thing being our landing README and the links to our documentation. So here in our README here, again, citations, suggestions, all the data is fully open, so anybody can access it, anybody can access it freely, but we please request that you acknowledge us if you do use this data in publication, because it helps us get the funding to continue to maintain and further develop the gallery. So here we go, data sets. So here on the landing page, we have listed the data sets that we consider fully complete. And so this means all of the data has been uploaded from most data sets have, image data all the way through numerical data. All of them have metadata, so you can fully understand what it is you're accessing. And so just to sort of give you an idea of what that looks like here, each data set has its own identifier. And so that'll start with a CPG number, and it'll have some tag, sort of, one or two words describing what that data set is. We also list a brief description to help orient you towards all of these different data sets that'll tell you basically whether it's, compound or genetic perturbation, what the cell line is, what the point of that data set was. Links to the publication that describe this data set, where you can dive in for more. Most of the data sets also have associated GitHub repositories that sort of curate more information that may be data analysis workflows. It may be things related to the publication. But again, links for you to dive even further into any of these data sets. And then briefly, the total size of the data set, and then either the image size or the numerical data size. So you have some idea of what you're getting yourself into if you're wanting to work with this data. What is the scale? So that's kind of the how to get the big picture overview of what's there in the gallery. But certainly it's 700 terabytes. You don't want to just download the whole gallery. So how do you find more specifically what you want? And for that, I will now show you into our documentation. So again, here is the link. Then this is our documentation. I think it's pretty comprehensive because I wrote it and I love documentation. But if you have, again, any further questions about the gallery that are not answered in the documentation, you can always email me, or in our GitHub repository, there is a tab called Issues. And here you can create a new issue. And it's a location to ask questions specific to the cell painting gallery, things that you would hope would be addressed by documentation, but you're not finding an answer to. So that is where to ask more specific questions. So within our documentation, this is maybe the, I don't know, least exciting part of the talk, but I think sort of the most important for understanding how to find data is talking about data structure. So all of the data within the gallery has very stereotyped data structure, meaning every data set has the same folders in the same organization, so that if you know, a specific piece of data that you want in one data set, you know exactly how to find it in another data set. I will point out here that this is, so I'm talking about folders. For those that have more technical experience, you may recognize this is an S3 bucket. So this is actually object storage. If you don't know what that means, it's okay. You don't need to know. This is, it is in object storage, but I'm sort of using buckets and paths interchangeably just so that's not a piece of confusion. So within the cell painting gallery bucket, again, I talked about how there are different project identifiers. Within each project, we have an additional nesting. Most data sets were produced at one location, and so that will be a tag sort of identifying that one location. Some data sets came from multiple locations, and so those will have the different sources listed as different nestings at this point. It can be anonymized. Again, if you have data you want to make public but don't want it public, you don't want it associated or named, we can publish data anonymously. And then within there, again, we have the images and the workspace divided. And so images are what you might imagine image data, workspace is sort of all of the numerical data. So that's sort of big picture, top level. And I'm going to talk you through sort of sub top level now, but rather than go through documentation, which I find more exciting than many, I'm going to actually show you one way to browse the data and what that might look like to browse on through. So here in our docs, we have browsing data on the cell painting gallery. And so the first thing listed here is that the cell painting gallery is indexed on Quilt. So Quilt is a company that that does lots of things, but we use to provide this public and easily browsable, like in your web console way to navigate through this gallery. So here we see, so we're at the sort of root of the cell painting gallery, and you see all of these different data sets. If we pick one to dive into, here JUMP is our largest data set, and you can see it had many sources as the identifiers there. Within that, there is images, workspace, and again, some projects have deep learning derived numerical data. Those would be in a workspace underscore DL. Almost all the other data sets will see sort of images and workspace. And if we click on images, so everything that, all of the data is generally organized first by batch, and then by plate, because these are, acquired on an assay, a plate, often 384 well plate that is imaged on a microscope, and a batch is generally, a stack of plates that were sort of handled on the same day or in a similar, unit of time workflow. So here within images, we see a list of batches. Within this batch, these are different folders describing the different kinds of images that we have. So the images folder is always going to be the images as they are raw, right off the microscope. There are some data sets that have sort of more major image handling that happens because of a specific workflow. And you can see here, like the Periscope data set, we have different images folders where we have sort of saved out some image intermediates so that one does not need to recreate the entire image handling workflow. But these are, again, these are all within the images folder. Then within the images folder, this will be a plate worth of images. And then everything below this plate identifier, this can vary depending on the microscope that it was acquired on. So sometimes there's another images folder, right? Like 3 different nested images folders. It's some redundancy, but it helps to keep everything very, again, organized and stereotyped. And so if we click, you can see there is some level of image preview that you can even get on Quilt, although it is not optimized for scientific image viewing. But you can see I've navigated my way all the way to an image. So that's sort of the path through the images and finding images and what that looks like. Also important to understand the workspace, the numerical data. So within so within every workspace, you will find a subset of folders that are in every project, and then additional folders that may be included. And we have, again, like all of this is described very thoroughly in our cell painting gallery folder structure in our documentation. So I'm not going to belabor all of the individual components because I've got more information there for you. But I do want to point out that generally what folks would be thinking of most accessing would either be metadata to help you understand what is the metadata associated with each of the images. Load data CSV is the CSVs as we feed them into cell profilers. So they associate actual paths to files and file names with the channel names that we have given them to describe them. So you know that, channel 4 is the RNA channel, for example, that will be described in a load data CSV. And then profiles, again, here organized by batch and plate. These are the profiling data, again, at different levels. So this is sort of, at its raw most per well basis, all the way through, normalized and feature selected to the negative controls here, as you can tell just by reading the file name. So lots of different lots of different profiles often, and you can select which one you would want through navigating through the profiles folder. So that's sort of how to find things and how to navigate around. So then you want to download data, right? Let's now talk about that. So because this is an AWS bucket, the most used tool, sorry, downloading from Silicon, here we go. So so we describe here kind of two different easy to use tools for downloading data. And so the AWS CLI, which is the command line interface, so it's going to be a tool that you will use in your terminal. Let's see if I can. And you can actually use the AWS CLI for both browsing, navigating as well as downloading. We have that, again, all described in documentation. So you can download the AWS CLI directly from AWS. They have tutorials. We link to that. But I'll just show you, for those that are not super familiar with using a command line, you don't even have to figure out how to generate your own download commands. It's kind of a neat thing about Quilt here that, for example, if I figure out that I want, let's say, this specific load data CSV here, up here in this tab, there is code tab with both a Python command and a CLI command. And so here, the Quilt has generated this AWS CLI download command for me. So I can just copy and paste that in. And this is saying, okay, AWS S3 CP is copy. This is the path to that load data CSV that I want to download. And this dot means download it in the location that I currently am navigated into. And there we go. It downloaded it for me. You don't have to have an AWS account to download things. If you do not have an AWS account, so I have one and my information is already saved in here, but if you don't have one, you can put --no sign request. And when we do that, that means it downloads it without recognizing that you have an AWS account or using an AWS account. So if you get an error in trying to download because you don't have an AWS account or don't want to create one, you don't have to. You can add this on. Another thing you can do that is helpful is if I'm wanting to see what would happen if I were to use a sync command or a download command without actually doing the download, you can enter dry run and it will, oops, that tells me it doesn't exist. So because that's not a specific file, there we go. If I tell recursive, it's going to look through that whole folder. And with this flag dry run, it's telling me, okay, if I ran that command, what would happen is it would download these 4 files for me. So it's a really helpful thing to know, if you have written out your download request correctly before actually triggering download, so you're not accidentally downloading things you don't want. So there's also, Quilt has a Python tool. So if that's more familiar and comfortable, they have docs on how to do that, how to use their tool to download data with Python. But really, it's really as simple as finding, browsing to the data you want, and then performing a simple download command. As we reach the end of my half hour here, I also want to point out you don't have to download the data. So we have linked here a couple examples of other workflows that access data directly from the gallery. Distributed Cell Profiler is a tool that we maintain that runs copies of Cell Profiler in Dockers using AWS resources. And in this example that we have here, it will actually even mount the AWS cell painting gallery bucket sort of as a pseudo file system. If these are words you don't understand, it's okay. The point is that we don't actually even have to download any data. It can mount and read the data directly from the gallery in this example workflow. Likewise, you can, this is a snake make pipeline. That, as this example, you can filter it to access based on just specific metadata that you want. So lots of examples. If you have other examples of workflows that access the gallery, let me know. I would love to add them to our docs to help people understand and have templates for other further workflows. One other thing that I briefly want to point out that we also have, two other things I wanted to point out. So Quilt also has, as I pointed out, has sort of tabular data previews, so you can see the tabular data in a preview in the gallery, which can help you just in browsing without having to download. They also have plot tools. So you can do really simple, very simple plotting and really simple data analysis, in the gallery, browsing through it in Quilt. And so here's some examples of that. Finally, we are in alpha right now, but coming soon, working on building up a whole suite of cell painting gallery tools. And so this has been a project that Ant Kumar has been working on in the Carpenter Singh Lab. And soon, we hope to have lots more tools directly querying and analyzing data, everything in the gallery. But one example that we have in alpha right now, you can actually search through all of the metadata. So if I type in a drug or a gene, it will search through the metadata and return to me, okay, in this project, it is found in this metadata. So I would know if I'm interested in how strychnine affects cells, I know that I will find it in this CPG 12 data set. And again, actively in development, so stay tuned for much more sort of simple in-browser handling of cell painting gallery data. So with that, I want to thank you for coming, listening. Also, if you're watching this real time or very shortly after recording, please do check out our Bridging Imaging Users to Image Analysis survey. It is a joint effort between many awesome groups where we're trying to understand the needs of imaging users and how those of us creating tools and doing image analysis can best help cross those bridges. So please check out the survey and help us. And with that, thanks everybody.